Okay, I want to start with this little video here.
DNA. We talk about it so much. It's the ultimate director for cells, and it codes for your traits. With a molecule that has a function like that, it makes sense that when you make another cell, like in cell division, you would also need to get more DNA into the new daughter cell. And that introduces our topic of DNA replication, which means making more DNA. First, let's talk about where and when. First, where? Well, if it's in a eukaryotic cell, it occurs in the nucleus. However, remember, not all cells have a nucleus, such as prokaryotic cells. They don't have a nucleus. Still, both prokaryotic and eukaryotic cells do DNA replication, but there's some differences between the two that this clip doesn't go into. Next, when? When does this happen? Well, a cell is going to need to do this before it divides so that the new daughter cell can also get a copy of DNA. To get specific, in a eukaryotic cell, that's going to be before mitosis or meiosis in a time known as interphase. I think DNA replication would actually make a great...
Okay, so, assuming that worked, we're gonna, just a reminder that let's see. So, here we talked about the cell cycle, and the major parts, the interphase, that's the biggest part of a cell's life. When the cell is doing the job of a cell, it's growing, it's getting ready to divide, and one of the main things about getting ready to divide is the S phase, or synthesizing, replicating, building, making DNA. And that's really what the rest of this chapter is about this process of DNA replication. Okay. We ended here looking at the actual structure of DNA, and you guys built a model about how you saw how it has a sound like a twisted ladder, with the sides of the ladder being these sugar and phosphate groups covalently bonded, and the rungs of the ladder are nitrogenous bases that are hydrogen bonded together in a very predictable fashion. Adenine always binds with thymine, guanine always binds with cytosine. Okay, so that's good background for where we're going to start our discussion of how does DNA actually replicate. Towards this part of the chapter, in terms of the historical aspects and people's contributions, we talked about Watson and Crick, we talked about Rosalind Franklin, about Chargaff, and about the transformation experiment. Mendelssohn and Stahl, Mendelssohn? I thought it was Mendelssohn. But Mendelssohn and Stahl, basically what they showed, and their contribution, is that they proved that DNA replication is conservative. You can read about the actual experiment here. Again, it's similar to this experiment here, Hershey and Chase's, where they're actually labeling molecules and then following them through these processes, right? In the Mendelssohn and Stahl experiment, they're labeling DNA and looking as it goes through different generations and is replicated. And what the gist of this is, we start over here on the right hand part of your screen. You see this two gray bars. What I suppose those two bars are. Those are the two strands of a molecule of DNA. And we see after one round of replication that there are two gray and two blue, right? So the gray are the original strands that are conserved through the first round of replication. And the blue is a new strand of DNA that was added to the old. So in that way, it is conserving the old, but also making some new. And that is the way that DNA replication is semi-conservative. here are some details of this process. You definitely want to know these enzymes, right? You know when you see ASE, it is an enzyme that is a protein responsible for catalyzing some type of reaction. These are basically all anabolic or building type of processes. We see ligase, polymerase, primase, and helicase, topoisomerase. Okay, so you should know those. Just memorize what the enzyme does and try to put it all together into a story of how DNA replicates itself. All right. Trying to think. Maybe I'll hop back to the video. We'll watch the video and then we'll come back to this diagram. Okay. This is the Amoeba Sisters. Many of you guys have probably watched these guys in the past. I really like them. They really break things down really well.
Still waiting for that to be invented. I'm going to introduce the key
Okay,
players
here are
of DNA
those
replication so that you
enzymes.
can get some background information. Now remember, these are just some major key players. There's a lot to this process. Many of the key players are enzymes. In biology, when you see something end in ASE, you might want to check, as it's very possible that it's an enzyme. Enzymes have the ability to speed up reactions and build up or break down the items that they act upon. So here we go with the key players. Helicase, the unzipping enzyme. If you recall that DNA has two strands, you can think of helicase unzipping the two strands of DNA. Helicase doesn't have a hard time doing that. When unzipping, it breaks through the hydrogen bonds that hold the DNA bases together. DNA polymerase, the builder. This enzyme replicates DNA molecules to actually build a new strand of DNA. Primase, the initializer. With as great as DNA polymerase is, DNA polymerase can't figure out where to get started without something called a primer. Primase makes the primer so that DNA polymerase can figure out where to go to start to work. You know what's kind of interesting about the primer it makes? The primer is actually made of RNA. Ligase, the gluer. It helps glue DNA fragments together. More about why you would need that later on. Now, don't feel overwhelmed. We'll go through the basics of this sequence in order. But remember, like all of our videos, we tend to give the big picture. There are always more details and exceptions to every biological process that we can't include in such a short video. DNA replication starts at a certain part called the origin. Usually this part is identified by certain DNA sequences. At the origin, helicase, the unzipping enzyme, comes in and unwinds the DNA. Here's the thing though, you don't want these strands to come back together. So SSB proteins, which stands for single-stranded binding proteins, bind to the DNA strands to keep them separated. And topoisomerase, I always have to slow down when I say that enzyme's name, keeps the DNA from supercoiling. Supercoiling might sound super, and it can be when you're trying to compact the DNA, but it's something that needs to be controlled during DNA replication. Supercoiling can involve an overwinding of the DNA, and you need the DNA strands to be separated for the next steps. Primase comes in and makes RNA primers on both strands. This is really important because otherwise DNA polymerase won't know where to start. In comes DNA polymerase. Okay, before we go on, remember how we said DNA has two strands? They're not identical, they complement each other. In our video that covers DNA structure, we talk about how the bases pair together with hydrogen bonds. The base adenine goes with the base thymine, and the base guanine goes with the base cytosine. These strands are also anti-parallel, so they don't go in the same direction. What do we mean by direction? Well, with DNA, we don't say north or south. We say DNA either goes 5' to 3' or 3' to 5'. What in the world does that mean? Well, the sugar of DNA is part of the backbone of DNA. It has carbons. The carbons on the sugar are numbered right after the oxygen in a clockwise direction. 1', 2', 3', 4', and 5'. The 5' carbon is actually outside of this ring structure. Now, you do the same thing for the other side, but keep in mind DNA strands are anti-parallel to each other. So let's count these, again clockwise after the oxygen. 1', 2', 3', 4', 5'. And the 5' is out of this ring. This strand on the left runs 5' to 3'. And the strand here on the right runs 3' to 5'. We'll explain why all that matters in a moment. So let's take that knowledge there and look at DNA replication here. In this image, I labeled the top original strand 3' to 5'. I labeled this bottom original strand 5' to 3'. That's the original DNA that is going to be replicated. DNA is unwinding here thanks to helicase. In this example, it will keep unwinding in this direction. Primase places primers. DNA polymerase is building the new strands. Now, the thing about DNA polymerase is when it's building a new strand, it can only build the new strand in the 5' to 3' direction, meaning it adds new bases to the 3' end on the new strand. See how it's being built in the 5' to 3' direction? This one is called the leading strand. But take a look down here. So DNA polymerase once again is building a new strand in the 5' to 3' direction. But there's a bit of a problem here. See, as DNA unwinds, because DNA polymerase can only build the new strand in the 5' to 3' direction, it has to keep racing up here next to where this unwinding is happening. You can see why then this new strand is known as the lagging strand. On this lagging strand, primers have to keep being placed in order for DNA polymerase to build. These fragments that result are known as Okazaki fragments. Primers have to get replaced with DNA bases since the primers were made of RNA. Ligase, the gluing enzyme, as I like to nickname it, has to take care of the gaps between Okazaki fragments, sealing them together. At the end of replicating, you have two identical double helix DNA molecules from your one original double helix DNA molecule. We call it semi-conservative because the two copies each contain one old original strand and one newly made one.
Okay, so semi-conservative DNA replication, as Michelson install showed us. I had to make some changes here to this video. There we go. Okay. This should look something like the one from the Omega Sisters video. We see all the same players. We see the polymerase. We see ligase, helicase, topoisomerase. The molecule that we're wanting to copy, you see on the right side of the screen. So this is any given chromosome. So, and if you think about it, each chromosome, the only difference between each chromosome is not the red part here. That's just a series of repeated phosphates and sugars. The only interesting or different or new part are the bases in the middle. Okay, they're shown here as the yellow and green pegs that are going across the middle. That's these, thymine, adenine, guanine, cytosine. The red on that video, on the diagram is shown as blue here, or it's just these sugar and phosphates. So, to make a copy of this, first thing we need to do is use this enzyme called helicase to break those hydrogen bonds between the bases. So break these bonds here shown in dashes. Now, if you imagine two strings of twine that are intertwined and you wanted to pull them apart, you would see that the top part, if you did this too quickly, the top part has under tension and it would bunch all up and make into a cluster. This topoisomerase, it unwinds the DNA in a more controlled fashion. So we have exposed these new bases, and they were stable when they were hydrogen bonded to another DNA base, but when they're not hydrogen bonded, they're very sticky and very reactive. They're wanting to react with another base. So to make sure that doesn't happen prematurely, there are these things called single-strand binding protein. They're placeholders, effectively. Polymerase comes in and we see that shown here associated with the sliding clamp. In this case, the DNA polymerase is moving from the screen left to the screen right in a 5' to 3' fashion. And what's it doing? It's adding in nucleotides. So it's adding into here a sugar phosphate and a base, and adding in the base that's complementary to the base that's on the old original strand of DNA. Okay, using those Charcap's rules. So if the polymerase finds an adenine, it's going to add a thymine. If it finds a guanine, it's going to add a cytosine. And it keeps doing this and adding them in. As you recall, the polymerase doesn't do this. It doesn't know where to get started. And so there is another enzyme called a primase that adds in a small, would have done it over here on screen left, it would have added in a little primer. Helicase gets started and then zips along 5' to 3'. We see that on the other, called a lagging strand, the primase here, shown this little blue ring, is added in these 3 nucleotides of RNA that are complementary to this DNA, and then a DNA polymerase will come in and it will move again 5' to 3' in this direction. Remember, these strands are anti-parallel. So the original strand here goes 3' to 5', while the complementary strand here goes in the other direction, 3' to 5'. And so when these DNA polymerases are working, they're going in opposite directions too, effectively. Helicase is opening. On the leading strand, it's a pretty easy task for the polymerase. It just waits for the helicase to expose new bases, and it just adds in the complement. On the lagging strand, it is a lot more difficult, because as the helicase opens the molecule going in this direction, then it allows the primase to come in, put a primer, and then it just seals in to the next one, but then it has to wait until more of the hydrogen bonds are opened up, more bases are exposed, and then, so effectively what you get is not a long continuous strand of new DNA like we see in the leading strand, but a whole bunch of these little fragments called Okazaki fragments. So, to make sure we go from a bunch of little fragments to one continual strand, there's a ligase shown here, comes in, it ligates or breaks open the RNA, and then there is another polymerase that comes in and seals that closed. Yeah. So that's the basics of DNA replication. Table 15.1, a good summary of these individual enzymes. You'll see. I'm not worried about the difference between DNA polymerase 1, 2, and 3. Just know that they all add nucleotides in and polymerize or synthesize DNA. Primase lays down the primers, polymerase lays down the DNA, and then we have ligase is what tapes together these individual little Okazaki fragments. This is words for the same thing, right? This is basically the same basic information in word form. Okay, now, we said all throughout our discussion of mitosis, the goal of mitosis is to make a new exact copy of our DNA. So when we look at our cell cycle, when we look at our cell cycle, the goal being to make an exact copy. However, is you can imagine this little enzymes that are inside of your nucleus and making copies of trillions of base pairs over and over and over again. It's a low probability that a mistake will be made, but they are made fairly regularly. So it's so critical that DNA copying happens in a really orderly fashion. The cell has their own basically editing enzymes. Okay, so DNA polymerase, in addition to making copies, will have some proofreading and repair functions. Okay, so in this case, we're proceeding. Which direction is the polymerase moving? It always moves from 5' to 3'. So here it encountered, right, cytosine, so it laid down a guanine, thymine, adenine, guanine, cytosine, guanine, adenine, thymine. But we see there are some problems with this, and it's starting to lay down. Oh my god, I hate this book sometimes. I know it's free, but so this, if we look at it, this should be complementary to the cytosine, this complementary to the thymine, which it is, the adenine, which it is, but then when we get to this G, it should be making a C, but it didn't. It added in this extra thymine, right? And this is a problem. So DNA polymerase will actually go in, right? It finds those mismatches, and it will cut out the newly synthesized strand, and then it fills in that gap using a correctly paired base. So it'll excise this thymine, and it will add in the cytosine. Very common mutation during DNA synthesis is called a thymine dimer. So it's actually the correct sequence. On the original strand we had 2 adenines, so on the newly synthesized strand we put 2 thymines, but rather than thymines hydrogen bonding to the adenines, they bond to each other covalently. It makes a dimer, which makes a little bubble, and it's just not the right structure, right? And so there's a little enzyme that rides along this kind of like a railroad track, and it detects this little bump. It stops, it cuts that out, and it adds in new thymines and makes sure that they hydrogen bond with the adenine. Very common. It happens in our skin cells when exposed to UV. We're just going to skip this telomerase stuff. Yeah, just going to skip it. So, what is the big deal with these mistakes? We'll call these mutations. Uncorrected replication errors result in mutations. So we have our original strand shown on the left. Okay. So in each of these we have the same AGC, GTA, CCC, TAC sequence. As we're about to see, this is kind of a little preview. The sequence of bases on a strand of DNA, the template strand of DNA, is what determines the sequence of amino acids in a protein. Okay, so we've been talking about how DNA, and one gene is a segment of DNA, and it codes for one protein. How does it code? This is sort of how it codes for the protein. 3 letters on a DNA strand code for one amino acid. And the sequence or primary structure of that protein is what determines how it folds up, and it determines its shape, and it determines its function. So it goes to show that if you change the sequence of bases in a DNA strand, it could change the sequence of amino acids. So SER, VAL, PRO, this is serine, valine, proline, tyrosine, these are amino acids. Okay, so if in here we have a mutation in this adenine, that would be the 1, 2, 3, 4, 5, 6th base pair. If it gets mutated and in the replication process, instead of adding an adenine, we end up adding a thymine, it doesn't have an effect on the protein because there's still a valine is transferred to the growing protein. However, right, if we have, let's see, where is this? Oh yeah. So here, the mutation is in the 7th. So the cytosine gets substituted for an adenine, and because we don't have the 3 letters CCC, we have the 3 letters ACC. Instead of bringing a proline, we bring a threonine to the protein. That's called a missense mutation. Nonsense mutation, right, is where instead of bringing an amino acid, it basically tells the cell to stop making a protein. And we're going to see that is going to have a big impact on a protein. So basically, when we talked about, we ended the last chapter 13 when we were talking about genetic disease and about how you can have a change in DNA which results in a funky protein which results in sickle cell, cystic fibrosis, maple syrup, urine disease. This is exactly what we're talking about here, right? We have a change to the protein, and that is caused by a change in the DNA. Friendship mutations is a really big effect on the protein. So we start out with our same protein, the serine, valine, proline, tyrosine. We end up with a totally different protein. Why? Because, let's see, it's hard to sort of see where this happens, AGC, GTA. So in this case, the T and the A are removed and the CCC shifts over. So rather than having GTA, CCC, we have GCC, which completely changes the entire sentence or the entire code that this gene has, and we end up with a completely different protein. So these mutations, replication is not perfect, errors happen, those are called mutations. They could lead to a change in this DNA sequence, which could lead to a change in the protein sequence. That's the point. Review here about compacting DNA. Here on the top is that double helix. It is that long linear strand of DNA. It is wrapped around a protein. So here, this double strand of DNA is shown just zoomed out. So we see that long strand, it's wrapped around a protein. The protein, look, you can see those multiple little proteins, secondary structures, tertiary, quaternary structures. This mix of green and blue and yellow and red things is shown down here as these little yellow balls. They're called histone pores. So this is how our cells compact this long strand of DNA into a compacted, duplicated chromosome. Okay. Now, very common technique that's used in the biology laboratory is to try to determine the sequence of bases in a strand of DNA. This is called Sanger sequencing. Okay, and I think the only thing I'm going to say about this Sanger sequencing is just a technique used to learn the sequence of bases along a strand of DNA. So it will tell you that there is a G in that guanine, adenine, thymine, cytosine, right? And that is the first step oftentimes in identifying a different species, identifying individuals, identifying whether somebody has a disease, because this can be traced back to what the protein sequence will be. Okay. Another common technique is called gel electrophoresis. Okay, so if we have made copies of our DNA and we want to visualize those copies, or if we have a long strand of DNA and we cut it into little pieces and we want to look at those pieces, we use gel electrophoresis to sort the fragments by size. Okay? Because we know DNA has a slight charge, we put it into this gel. It's a horizontal gel. You put it in here, you apply a electrical current, and it pulls the fragments through the gel, and it allows the smaller fragments to move through the gel more quickly. The larger fragments get left behind. Okay. So Sanger sequencing determined the sequence of bases along a strand of DNA. Gel electrophoresis will allow you to visualize how many fragments and what the size of the DNA fragments are. Okay, so that's chapter 14. And it's a big one. This is important stuff, particularly thinking about the structure of DNA, right? About what these nucleotides are made up of: a sugar, a phosphate, and a base. But don't lose sight of this right here. This sugar, phosphate, and base is the same as this sugar, phosphate, and base is the same as this sugar, phosphate, and base. It's all the same kind of thing, right? DNA fragments are anti-parallel. We see 3' to 5' on this side, 5' to 3' on this side. They're held together by hydrogen bonds, and when these bases really are what is unique about each individual chromosome, the sequence of those bases. When the cell wants to make a copy of itself, it wants an identical copy of all of its chromosomes. It uses this semi-conservative method to make copies of the old strand, making new strands. Okay? So, thanks.